Mustafa Sahin's journey in neuroscience began in mapping neurotransmitter and peptide receptors in inflammacortical pathways as an undergraduate researcher and a biochemistry major at Brown. He completed the MD PhD program in neurobiology with Susan Hockfield at Yale, where he was elected to AOA, Medical Honor Society. He pursued pediatrics at CHOP and ultimately a child neurology residency at Boston Children's. And in the midst of that very busy, demanding training program, he formulated a successful K-Award project that he completed over the next few years with developmental neurobiologist Michael Greenberg at Harvard. That important work culminated in a series of 3 papers in Neuron on fundamental mechanisms of neuronal and brain development. He has always embraced the synergy between studying neurogenetic diseases in children and exploring fundamental mechanisms of neuronal and brain development. Tuberous sclerosis became a focus of his work, but he continued to study spinal muscular atrophy as well as Fragile X and other neurogenetic disorders associated with autism phenotype. Among many national and international awards, he received the John Merck Scholar Award and the Jacobs Ladder Norman Sanders International Research Prize from the Hospital of Sick Children in Toronto. Mustafa Sahin has embodied a commitment to the synergy of collaboration. He mentored at least 34 medical and graduate students at Harvard, MIT, and beyond. He mentored at least 27 postdocs and faculty, including 5 KOA recipients and 4 R25 recipients. With a vision of the possibilities of translational research for families affected by severe neurodevelopmental disabilities, including tuberous sclerosis, in 2013, he created and directed the Rosamund Stone Zander Translational Neuroscience at Boston Children's. He was later appointed as professor of neurology and endowed chair in neurology. In our brief time together, I hope that we will benefit from a brief glimpse at how a pediatric neurologist and developmental neurobiologist envisions translational neuroscience in the service of our patients and the quest for understanding fundamental mechanisms in brain development. Welcome, Mustafa. It's great to have you here.
Thank you so much, Dana, and thank you for the invitation and the very kind introduction. I'm going to share my screen. All right. Good morning, everyone. What I'd like to do this morning is to tell you about some of the translational work we've been doing in my lab and with our collaborators on the mTOR pathway, a treatable target in the disease tuberous sclerosis. I'll start with my disclosures because of the translational nature of our work. I work with a number of biotech and pharma companies that are listed here, and I will discuss the use of sirolimus, vigabatrin, and besimglutarant in clinical trials. I want to give you a broad introduction about where I think we stand in neurodevelopmental disorders today. And I'll use autism spectrum disorder as the example. Due to advances in genetics and genomics, we now believe that there are about 400 to 1,000 different susceptibility genes underlying autism spectrum disorder. This has been wonderful in terms of a huge number of publications in this field and identifying genetic causes. However, it obviously creates a potential handicap in terms of treatments, identifying treatments. One can think of a number of different scenarios on how we can approach this. One can imagine a broad spectrum intervention that works for all genetic causes of autism. But due to basic science work in diseases such as Fragile X and tuberous sclerosis, which shows that they're essentially diametrically opposite to each other in terms of hippocampal physiology, for instance, this may not be feasible. In fact, some interventions, some pharmacological interventions that help a certain group of patients may be harmful in a different group of patients affected with autism spectrum disorder. On the other hand, we may consider a scenario where we have to develop a different intervention for every single genetic variant, which is an incredibly resource and labor-intensive scenario that I don't think we are ready for yet. So many of us in the field believe that there might be subcategories of autism that share a convergent focus either at the circuit level or at the cellular level. And these subcategories may respond to the same interventions. Along those lines, we've proposed that abnormal mTOR activation either too much activation of the mTOR pathway or too little activation of the mTOR pathway may be a common target in autism. Now, along those lines, we have focused on a disease called Tuberous Sclerosis Complex as a model to study not just autism, but also epilepsy. And here are some of the reasons TSC comes to the forefront. 90% of the patients with TSC are affected with epilepsy sometime in their life, and 50% of the patients with TSC are affected with autism spectrum disorder. Importantly, many of the patients with TSC will be diagnosed before birth or at the time of birth due to the presence of heart lesions, which I will talk about. This gives us a window of opportunity for intervention early in life. The cellular mechanisms aberrant in TSC, the mTOR pathway or mechanistic target of rapamycin pathway, are beginning to be understood from a number of perspectives, not just from neuroscience, but from transplant biology, from cancer, from basic cell biology in general. And importantly, we have FDA-approved inhibitors of this pathway that can be repurposed in either preclinical or clinical trials to interrogate certain hypotheses. So the combination of these four factors makes tuberous sclerosis a unique opportunity in this field. I'm going to give a relatively short background on tuberous sclerosis. Many of you are familiar with this disease, I assume, but it's a genetic disease that causes benign tumors in the brain, eyes, skin, kidneys, and the heart. It usually presents with seizures. That's why child neurologists like myself get involved, and there's a high incidence of intellectual disability and autism. The genes have been known since the 1990s. TSC1, also known as Hamartin, and TSC2, also known as tuberin, are the causal genes. And if you have a heterozygous mutation in either one of these genes, that is associated with the presence of tuberous sclerosis disease and its signs and symptoms. The incidence is thought to be about 1 in 6,000 to 1 in 10,000. So we expect about 50,000 people in the United States, about 1 million people worldwide affected with this disease. The diagnosis of TSC has been, until recently, based on clinical criteria. This is the updated clinical criteria published in 2021 by International Consensus Conference. There are 11 major features and 6 minor features, and having 2 major features or 1 major feature plus 2 minor features is sufficient to give you the definite diagnosis of TSC. Having just 1 major feature or 2 or more minor features gives you the possible diagnosis of TSC. I just want to highlight the major features. There are 4 features on the skin: hypopigmentic macules, facial angiofibromas, ungual fibromas, and the shagreen patch, usually in the back of the individual and the lumbar area. There are 4 CNS manifestations: retinal hamartomas, cortical tubers, suprapendimal nodules, has to be more than 2, suprapendimal giant cell astrocytomas or SAGAs. And then finally, there's a cardiac manifestation, the rhabdomyomas, lung manifestations, lymphangiolymatosis, and then angiomyolipomas, usually in the kidneys. Importantly, if you just have LAM and angiomyolipomas, that is not sufficient to give you the full diagnosis of tuberous sclerosis. And an important aspect that was recently added is that pathogenic variants in TSC1 or TSC2 are sufficient for the diagnosis of TSC. So you don't have to fulfill all the clinical features. A genetic diagnosis can be sufficient. Along those lines, in terms of genetic testing, by sequencing of the gene and looking for deletions using MLPA, which is multiplex ligation dependent probe amplification testing, you can identify the pathogenic mutation in 75 to 90% of the individuals with a clinical diagnosis of TSC. That leaves behind about 10 to 25% of the patients where we don't identify the mutation by testing blood DNA. We don't identify mutation in either TSC1 or TSC2. So this has led to search for other potential causes of TSC. And we work with David Kwiatkowski at Brigham and Women's Hospital, who used next generation sequencing and showed that many of the patients in which no mutation was previously identified by conventional testing actually are mosaic for TSC1 or TSC2 pathogenic mutations. So those seem to be the major cause of the NMI or no mutation identified patients. In general, patients in which we don't identify the pathogenic mutation in the blood are milder cases of TSC disease. Now, we know the genetic cause of this disease. At the cell biological level, TSC turns out to be a disease of cell size dysregulation. This on the left is a brain tissue histology showing the presence of these giant cells. These cells can be 10 times the size of a normal neuron and are pathognomonic for this disorder. And it turns out in any organism in which you get rid of TSC1 or TSC2 function, you get enlargement of cell size and organ size. Here's a fruit fly eye. The wild type eye is on the left. The TSC affected eye is on the right. You see the whole eye is bigger. Ommatidia that make up the fruit fly are bigger. And the hair cells, if they're missing the TSC1 gene, are larger. And this pathology is true for all the manifestations we see in the TSC patients. Most of the time in the hamartomas, we see loss of the second allele. So you see the cells that are null for TSC1 or TSC2 that are associated with these hamartomas. In the brain, this presents as cortical tubers. As you can see on this FLAIR imaging, the white signal represents cortical tubers. On average, a patient with TSC will have 20 to 30 cortical tubers. On the left side, you'll see a postmortem specimen where you see thickening of the cortex as well as lack of myelination underneath that cortical tuber. This patient also happens to have a subarachnoid giant cell astrocytoma shown by the lower red arrow. Subarachnoid giant cell astrocytomas are seen in only a subset of patients, somewhere on the order of 5 to 15%. They can cause ventricular obstruction and hydrocephalus. The median age that they seem to be detected is around 9 years of age, so they are relatively rare in the newborn period and rare after the age of 20. But this is the reason it's been recommended to do a follow-up MRI every year on patients that have subarachnoid nodules. If they don't have the subarachnoid nodules, we do MRIs every 3 years. Next slide shows what we're trying to avoid, which is a subarachnoid giant cell astrocytoma at the foramen of Monroe that causes obstruction of CSF flow and hydrocephalus, and this is a neurosurgical emergency. We also detect cardiac rhabdomyomas by ultrasound, and this is an early sign of tuberous sclerosis. They can develop in utero, especially in the second trimester. Post-birth, they almost always regress spontaneously, often completely disappearing as the child grows older. Wayne Turesky, one of our cardiology colleagues here at Boston Children's, had done a study about 20 years ago when he collected from a multi-center study about 100 patients with cardiac tumors in the newborn or prenatal period. And the frequency of having TSC in this 100 patients was about 70%. But if you had multiple cardiac tumors, the chance of having TSC went up to 95%. Those with single tumors, which we sometimes see and follow, the chance of having TSC is about a quarter. Skin involvement is an important manifestation of tuberous sclerosis in terms of diagnosis. 90% of the patients will have hypomyelitic macules that are best seen with a Woods lamp examination. 70 to 80% will develop facial angiofibromas, usually starting around 2 to 6 years of age, but increasing with puberty. Shagreen patch, which is a collagen-filled patch, is often on the back of the flank. And then in about 15 to 20% of the cases, starting in adolescents and adults, usually you see angle fibromas. Here are some images of hypomyelitic macules that can be sometimes easier to see versus harder to detect, depending on the background skin color of the individual. They can be anywhere on the body. And then facial angiofibromas are rather subtle in young individuals, as you can see on the right, whereas they can become quite disfiguring as the individual grows older. Shagreen patch, this collagenoma, is usually seen in the back of the flank, but they can be seen anywhere on the body, as shown on the ankle of this child. Now I'm going to switch gears from the clinical manifestations to the cell biology of this disease. As I mentioned, mTOR pathways, particularly involved, and this was discovered by 5 different labs around 2002, 2003 around the world. It turns out the protein synthesis and cell growth in the cell is under the regulation of 2 critical proteins, a GTPase called Rep and a kinase called mTOR, which stands for mechanistic target of rapamycin. And when these 2 proteins are active, they turn on through phosphorylation a set of protein machinery, resulting in increased protein synthesis and cell growth. TSC1 and TSC2, on the other hand, form a protein complex. Together, they inhibit Rep. It turns out TSC2 is a gap activating, GTPase activating protein, or a gap for Rep. And so it puts a brake on Rep activity. And its normal function of this complex is to keep protein synthesis and cell growth under check. As you can imagine, if you are missing either TSC1 function or TSC2 function, Rep and mTOR become hyperactive, and that results in increased protein synthesis and cell growth. Cells have taken advantage of this pathway in several ways. So various growth factors, including insulin, IGF, BDNF, brain-derived neurotrophic factor, all increase cell growth and protein synthesis through activation of a kinase called AKT, which inhibits this pathway by phosphorylating TSC2 on a certain set of residues. I should highlight P10, another important phosphatase that's been involved both in cancer and on autism, is also intimately involved in this pathway. On the other side, energy level in the cell does the opposite. So if you break down your ATP to AMP, that activates a kinase called AMP kinase, which phosphorylates TSC2 on a different set of residues and activates this protein complex, putting a brake on mTOR activity. So a starving cell doesn't continue to make protein and continue to grow. So the connection between TSC and mTOR, which happened around 2002, has really been seminal in terms of the work that has happened since then. It has really changed the landscape of what's been happening in the TSC field. So mTOR inhibitors, which give the kinase its name, were initially identified. The first example was rapamycin, which was initially identified on Easter Island in the Pacific Ocean. This chemical structure is shown here. It was identified as antifungal at first. It turned out to be an immunosuppressant that has been used in solid organ transplants for decades now. But rapamycin and its analogs that have been generated since then have been very important for tuberous sclerosis as well. So the first indication came from David Franz in Cincinnati, who used rapamycin in patients with tuberous sclerosis and subarachnoid giant cell astrocytomas. This led to another study using everolimus, which is very similar to rapamycin, but different by just the presence of one methyl group. And this was a clinical trial of only 28 patients with subarachnoid giant cell astrocytomas with tuberous sclerosis. It was a single-site study, and investigators showed that in all 28 patients, use of everolimus led to either shrinkage or stoppage of growth of the subarachnoid giant cell astrocytoma, and this was sufficient for the FDA to approve this drug for subarachnoid giant cell astrocytomas in tuberous sclerosis around 2010. The second indication turned out to be renal angiomyolipomas, which I will not speak more about, but a third indication was epilepsy. And this required a larger phase 3 trial of everolimus in TSC patients. About 340 patients were involved in this double-blind, placebo-controlled trial. This was also positive and led to approval of everolimus in TSC patients with epilepsy in 2018. One area where we have not been successful in showing efficacy is autism, and this is an area that my lab and our collaborators have been working intensely on. One of the first studies we did in autism in TSC was to look at the true incidence of autism in TSC patients. In retrospective studies up to that point had suggested rates ranging from 17% to 80%, based on whatever population was being studied. So Shefali Jesti, who was a chief resident at the time, collaborated with our colleagues in the United Kingdom who had babies born with TSC phenotyped using ADAS, the gold standard for research testing in autism. This was done at yearly intervals, starting at age 2 up to age 5. And what Shefali showed by her analysis with the UK data was that about 50% of the patients have either full-blown autism or the broader autism spectrum disorder criteria fulfilled, while the other 50% did not seem to fulfill the criteria for autism spectrum disorder. So one question we get asked a lot is whether autism symptoms in TSC are similar to autism symptoms in non-syndromic autism in patients. And Shefali looked at this carefully when she started her own lab at UCLA. She used this subscores using ADAS profiles and in TSC patients versus non-syndromic autism patients. Purple line shows the TSC patients with autism, and orange line shows the non-syndromic autism population scores. And as you can see, there's a remarkable overlap in terms of the subscores and on these different scales. So, typically there's a lot of convergence between TSC autism and non-syndromic autism. Then the question becomes, what causes autism in TSC? The first couple of studies focused on cortical tubers. Patrick Bolton's group in United Kingdom, again, had an important study published about 20 years ago using 1.5 Tesla MRI and identifying the presence of bilateral temporal lobe tubers as a necessary but not sufficient factor for the presence of autism symptoms. As you know, we now use 3 Tesla MRI, and almost all the patients with TSC will have some lesions in temporal lobes, which suggests that this may not be as specific a correlation. And several other studies using different cohorts failed to show a similar correlation in temporal lobes. Others have implicated tubers in the cerebellum or the frontal lobes. So the connection between tubers, tuber load and location and autism is still needs to be explored further. When I started my lab around 2005, we focused on the hypothesis that TSC1, TSC2 regulate neuronal connectivity and function through the mTOR pathway, and abnormalities in neuronal connectivity and function, regardless of the presence of tubers, may be playing an important role in manifestations of TSC, including epilepsy and autism. So over the years, we've looked for the function of TSC proteins in neurons and also non-neural cells in the CNS, and identified the roles for TSC in axon specification and guidance in neurons, in mitochondrial dynamics in neurites, in myelination, especially in a non-cellular autonomous way by TSC deficient neurons affecting oligodendrocytes surrounding them. And we also studied TSC's role in epilepsy, cerebellar circuitry, and circadian rhythms, because sleep abnormalities are a major manifestation of tuberous sclerosis. What I'd like to do today in the next 25 minutes or so is to focus on our work in epilepsy and cerebellar circuitry. Starting with epilepsy, as I mentioned, 90% of the patients with TSC will develop seizures sometime in their life. And remarkably, almost all the mouse models that we and others in the field have generated show seizures as one of the major manifestations. You can see on the left column the different Cree lines that have been used to delete the TSC1 or TSC2 genes in different regions or different cell types in the brain, and all of them have seizure phenotype. We started working with SNAPs in Cree as a way to delete TSC1 gene in post-mitotic neurons. This work was published in 2007, 2008. And what we found was working with David Kwiatkowski, who developed this model. These mice died from seizures, which started around 3 weeks of life. They all died from seizures by about 60 days of life, and they showed electrographic as well as clinical manifestations of seizures. We decided to treat these mice with rapamycin based on the scientific premise I already shared with you, that the mTOR pathway is hyperactive in these mice. And we used both rapamycin as well as everolimus. Rapamycin is shown on the left, everolimus is shown on the right. We started treating these mice at day 7 with the mTOR inhibitors. And vehicle-treated mice died from seizures, as I mentioned, by day 60. If you use rapamycin to treat these mice and kept them on rapamycin, 90% of them survived and they did not display any seizures. If you stop the treatment around day 30, about a month later, they started having seizures and they started showing early mortality. And we saw very similar results with everolimus as well. So this work and several other laboratories doing similar trials in different knockout models of TSC culminated in this everolimus trial that I mentioned earlier. This was led by Jackie French and was published in Lancet in 2016 and was a successful trial, as I mentioned earlier, and led to the FDA approval of this compound for epilepsy. I want to share with you one slide from that paper. This is the response rate of the patients as defined as greater than 50% reduction from baseline frequency. So we're not talking about becoming seizure-free. We're talking about going from, let's say, 10 seizures a day to 4 seizures a day. That's a positive response. And in the placebo group, the positive response rate was 15%. In the low-dose everolimus group, response rate was about 30%. In the high-dose everolimus group, the response rate was about 40%. And this is far from the 90 to 100% efficacy that I showed on the previous slide in the mouse models of TSC. So this begs the question why this compound that is mechanistically very appropriate for TSC patients does not result in 100% efficacy and seizure freedom. And what can we do to improve seizure control in TSC patients? So we've been looking for other mechanisms. We wanted to develop an epilepsy model of TSC using mice for our translational studies. The previous model that I showed you earlier is very fragile, and the mice do not tolerate anesthesia. They died through, if you try to anesthetize them, to put EEG leads on the head. So we developed a hypomorphic allele of TSC2, again, working with David Kwiatkowski. Instead of going to 0% TSC2 expression, we have about 8% of the TSC2 expression left, and these mice turn out to be much more vigorous. They tolerate EEG placement, and they also still show 100% penetrance of hyperactivity and seizures. We have a 24-hour video EEG monitoring unit in our animal care facility where we can record from 16 cages at the same time using a transparent cage, a receiver plate that records the EEG, and a night vision camera. Using this setup, we did a study of the different potential small molecules that might be helpful for TSC knockout hypomorphic mouse model that we developed. The first hypothesis we wanted to investigate was important finding by Mark Baer and his colleagues at MIT. They had shown that TSC and Fragile X might be opposite from each other, looking at long-term depression in the hippocampus. And what they had shown that is that long-term depression, which is a synaptic plasticity measure in the hippocampus, is increased in the Fragile X model. Fragile X, as you know, is a relatively common intellectual disability. They looked at the TSC2 mouse, which shows actually a reduction in long-term depression. So these are diametrically opposite effects. And if they cross the FMR1 knockout to TSC2 knockout, using a double heterozygous, that cross looked very much similar to the wild type. Based on this, they proposed that mGluR5, metabotropic glutamate receptor 5 inhibitors or negative allosteric modulators, might be helpful in Fragile X syndrome, whereas activation of the mGluR5 receptors could be helpful in tuberous sclerosis. This was a really important finding, and we wanted to validate these results. One thing that was concerning to us about this publication was that TSC2 heterozygous mice that was used in the study do not show seizures, whereas 90% of our patients in the clinic do have seizures. So we wanted to use a mouse model that actually has spontaneous seizures and test the effect of these compounds, mGlu5 positive allosteric modulators or activators in our mouse model. So we actually did this study. At the baseline. Our TSC2 knockout mice have seizures, as you can see here, and we published it in 2008. I'll show you just two brief panels from that paper. So the first thing we did was to use a compound from Roche that activated the mGlu5 receptor. What we saw was that our animals, seen at the left bottom, are hyperactive at baseline. This is an open field test, and every time the animal moves, you see these black lines forming. Our vehicle-treated knockout animals are hyperactive at baseline, but if you give them the compound that activates the mGlu5 receptor, they become remarkably more hyperactive. But more concerningly, seizures were also worsened by the treatment with this mGlu5 activator. As you can see, at baseline, our knockout mice, or the KC plus column here, you treated with vehicle has some seizures, 1 to 2 seizures per 2 hours. But if you give them 1 dose of the activator, they have markedly more frequent seizures, and they actually die from seizures in most cases. So this was quite worrisome to us, and led us to ask whether the opposite pharmacology, inhibition of the mGlu5 receptor, could actually be helpful in our TSC mouse model. So we used CTEP, which is an inhibitor or negative allosteric modulator of the mGlu5 receptors. And we looked at both hyperactivity and the seizure phenotype. As you can see in the left bottom, the frequency of the open field movements were significantly reduced after NAM treatment. CTEP also reduced the seizure frequency if used chronically. Compared to the vehicle, the number of seizures per 2 hours were reduced to about half. So these findings, as well as some biochemical data that we collected in that paper, led to a clinical trial that's ongoing right now. It's led by a Swiss pharmaceutical company called Noema. It's a clinical trial with an mGlu5 negative allosteric modulator called basimglurant, and it is started both in the United States and throughout Europe, and is ongoing. And hopefully, we'll have results from that study to see if our findings from this mouse model has some implications for clinical management of seizures in TSC patients. One of the other models that we would like to utilize in TSC investigations is human-induced pluripotent stem cell-derived neurons. We have an Intellectual and Developmental Disabilities Research Center at Boston Children's, and one of the cores is called the Human Neuron Core, where we can take either blood or skin samples from patients seen at our hospital or anywhere in the world, to be honest, and we can turn them into stem cells and take those stem cells and develop different types of neurons, in this case, excitatory or inhibitory cortical neurons, dopaminergic neurons, spinal motor or sensory neurons. And we have some small molecule libraries available for screening, up to about 30,000 compounds that we can screen in our facility for different phenotypes. So using this core facility, we've been studying tuberous sclerosis. This is a work led by Kellen Wind, another child neurologist funded by a K08 grant in the lab. What he did was to take iPSCs, induced pluripotent stem cells, from TSC patients shown in the middle. He induced a second mutation in the wild-type allele of that TSC2 patient iPSCs. He also corrected the TSC2 mutation in the patient. So we have an allelic series of wild-type, heterozygous, and null cells. As you can see, the null neurons are much larger than the other two phenotypes. He looked at the cell body size. There's an increase in a gene dosage-dependent way in the cell body size. Neurite length is also increased, and these are both phenotypes that we have seen in the mouse and rat neurons before, and we've now confirmed it in human neurons as well. If you have less expression of the TSC2 protein, both cell body size and neurite length goes up. But more importantly, Kellen was interested in looking at the electrophysiological properties of these neurons in culture. So he used multi-electrode array, where you plate the neurons on a dish with electrodes underneath, and you can record from the neurons in that well. And depending on the firing, you can see the presence of these black lines, and each on the left y-axis, if you will, is the presence of the electrodes in that well. And as you can see, the wild-type cells fire with some frequency, and sometimes they all fire together, showing some synchrony with, as you can appreciate, with the presence of these dark black lines going through all the electrodes. The wild-type cell, the heterozygous cells seem to fire roughly the same frequency, but they seem to have a bit more synchrony. But at the bottom is the presence of the firing rate from the null neurons. I should say these are excitatory neurons using a specific differentiation protocol. These null neurons fire more frequently, but they also fire even more synchronously. And you can quantify each one of these parameters. The firing rate is shown on the left graph. The red line is the TSC2 null neurons firing. That's much higher than the heterozygous and wild-type cells. And then if you look at the synchrony index, heterozygous cells are somewhere in between. The null cells are firing with highest synchrony, and the wild-type cells are at the bottom. We then asked the question, can we change this firing property using an mTOR inhibitor? So Kellen treated these cells with a low dose of rapamycin. And he showed that you can reduce the firing rate significantly back to essentially wild-type levels using rapamycin treatment. Importantly, if you wash out rapamycin, the firing rate comes back to the abnormally high firing rate that we see prior to treatment. So maintenance of mTOR inhibition seem to be required for this effect. This gives us a platform to test different compounds now. Before giving it to mouse models or to patients, we can actually look at the effect of small molecules or other interventions in this in vitro platform. And we've started doing that, doing phenotypic screens in TSC deficient neurons in a high throughput way. And we have identified some new compounds that are in the heat shock machinery pathway that seem to affect mTOR activity, as well as certain phenotypes such as cilia development in the TSC deficient neurons. So hopefully more to come from this kind of screening in the future. I'm going to very briefly turn to our work next on the cerebellum. Now, why did we start studying the cerebellum? The main reason was that Peter Tsai, who is also a child neurologist, who joined my lab as a postdoctoral fellow, was particularly interested in behavioral neurology, and he had a particular interest in the cerebellum. So his K08 grant application was focused on the role of cerebellum in TSC. Previous studies, which I don't have time to go into in depth, has suggested that individuals with autism can have abnormalities in the cerebellum, both using MRI or postmortem studies. This is one of the most common findings in the autism literature. An important autopsy collection of studies have shown that loss of Purkinje cells in the cerebellum is one of the more common findings in the autopsy specimens of individuals affected with autism versus typically developing controls, for instance. And then a study done by Katalin Propoulos, a psychologist who was at the time at Boston Children's, looked at newborns with hemorrhage in the brain during the neonatal period. And when she studied those with isolated cerebellar hemorrhage, 30% of those patients, when followed sequentially, developed autism spectrum by the age of 3 years. So these type of studies made us interested in studying cerebellum. There's actually quite a bit of data on cerebellum and autism in TSC. About 30% of the patients with TSC have cerebellar lesions on MRI, even though they don't have any cerebellar findings on bedside testing. Purkinje cell loss has actually also been detected in postmortem studies of TSC cerebellum by one study from Mike Gambello's lab at Emory. There have been two cohort studies looking at the correlation between cerebellar pathology and autism, and they've shown that tubers in the cerebellum is highly correlated with autism in patients with TSC. And then finally, an interesting study by Harry Shigani looked at PET imaging to look at metabolism in the brain of TSC patients with and without autism. And he used 2DG PET to look at the metabolism in different parts of the brain. And his finding was that in those patients with TSC and autism, there was hypermetabolism in the deep cerebellar nuclei. Just to remind everyone, deep cerebellar nuclei are inhibited by the cerebellar cortex. So this hypermetabolism or hyperactivity in the deep cerebellar nuclei indicates a hypofunction of the cerebellar cortex. So we wanted to study whether we could model this dysfunction in the cerebellum by knocking out the TSC1 gene just in the Purkinje cells of the cerebellum. So the way this cerebellar cortex is organized, all the cells in the cerebellar cortex end up sending synapses to the Purkinje cells, and the Purkinje cells are the only output cell of the cerebellum. So Peter decided to test this hypothesis that cerebellar dysfunction contributes to the pathogenesis of autism symptoms in TSC using a mouse model. I wanted to highlight this paper from Michael Gambella, where they had studied Purkinje cells pathology in TSC patients, and they showed that Purkinje cell density is reduced in TSC patients compared to controls. So what Peter did was to delete the TSC1 gene in Purkinje cells and looked at the behavior of the mouse model. I'm going to go through this slide relatively fast, but this is using a 3-chamber social approach test. What you do in this test is to put the mouse in the middle chamber of a 3-chamber plastic apparatus. On one side of the chamber, there's a live mouse under a cage. On the other side, there's just an object, and you give the mouse the potential to interact with a live mouse versus an object. And most strains of mice are social, and they like to explore the animal, the novel animal. So we did this using our control litter mates, heterozygous litter mates, and null mutants, which are missing the TSC1 gene in just the Purkinje cells. And as you can see in the gray bars, the control litter mates spend more time interacting with the animal than the object, and the time investigating is shown in the bottom graph. There's a strong preference to interact with the animal than the object. However, our heterozygous and null mutants, shown by the blue and red bars respectively, don't show that preference. They spend an equal amount of time with the animal as the object. This is a relatively rough measure of social approach preference in the mouse model. So these data suggested that missing the TSC1 gene, either one or both copies of the TSC1 gene just in the Purkinje cells of the cerebellum, were sufficient to give you an impairment in the social interaction in this mouse model. We also looked at cognitive flexibility using a water T-maze, which is another sort of crude measure of cognitive flexibility. You teach the mouse the location of a platform in a water T-maze. They learn the location of this platform, let's say on the left side of the arm of the T-maze, and then you change the location to the other side, to the right side. The first phase is just learning the initial location of the platform, which is a hippocampal task, and our mouse model did not show any deficits in that first phase. However, the second part requires the animal to forget the original location and learn a new location, and that requires the cerebellum. And our knockout mice, shown in the red bars, had a significant deficit on day 1 of this reversal learning paradigm, suggesting cognitive inflexibility, another core feature of autism that we see in our patients. So what Peter did was to take this mouse model and with the phenotypes that he identified, treat the mice with rapamycin starting from early on. He started the treatment at postnatal day 7, essentially the same time as the deletion of the gene is happening, and asked whether he could look at the Purkinje cell viability as well as some of the behavioral phenotypes of these mice. I did not mention this earlier, but similar to what's been reported in the TSC patients, there's actually a reduction in Purkinje cells when you delete the TSC1 gene in the mouse cerebellum as well. The graph on the top right shows that in the mutants, we see a reduction in Purkinje cell number, which is abrogated if you treat the mice with Rapamycin starting at postnatal day 7. But more importantly, we also showed an improvement in the social approach phenotype compared to vehicle-treated knockout mice, which did not show a preference for the animal versus the object. When you treated the mice with Rapamycin, they spend much more time interacting with the animal than the object. So this suggested to us that mTOR inhibition was sufficient to reverse this important social approach phenotype. While we were excited about these findings, we were bothered by the fact that we had started the treatment very early in life of this mouse line at postnatal day 7. What we wanted to do, which we thought would be more translation relevant, was to allow the animal to show some symptoms of this behavioral deficit and then start the treatment. So what Peter did was to treat the mouse model at different time points using Rapamycin. And this, since this is already published, I'm going to just summarize the data. He waited for 6 weeks and then treated the mice with Rapamycin for 4 weeks and looked at the behavior, or he waited 10 weeks and started the treatment for 4 weeks and looked at the behavior. And what we found was that if you start the treatment at 6 weeks, you can rescue the social approach phenotype, but you can no longer rescue the water maze phenotype or the grooming phenotype, which are both sort of repetitive, restrictive activities, if you will. So the two major features of autism-like behavior in this that we saw in this mouse model seem to have different sensitive periods to treatment with Rapamycin. And importantly, if you waited for 10 weeks, you can no longer rescue the social phenotype, social deficit either. So there seem to be relatively early sensitive periods that require the mTOR inhibitor treatment to start. So why does this matter? Obviously, this has important implications for basic science of what these autism-like behaviors in mice mean in terms of connectivity, circuits, and sensitive periods for treatment. But more importantly, on the clinical side, this has implications for about when we might be able to treat different cognitive and behavioral manifestations we see in our patients. The reason I'm highlighting that is that we had actually simultaneously run a clinical trial of mTOR inhibitor, in this case, everolimus, in TSC patients, and we had focused on neurocognitive features as the primary endpoint. This was a study done with Darcy Kruger at Cincinnati. In this 2-site study, we did 47 patients from 2 sites, ages between 6 and 21, randomized placebo-controlled trial looking at neurocognitive testing at baseline 0 and 3 months. I'm sorry, 0, 3, and 6 months. And we published this paper a few years ago. And what was disappointing is that we did not see an improvement in patients treated with everolimus versus the group treated with placebo in terms of the neurocognitive outcome measures. And this begs the question, in the light of our findings in the mouse model, whether the age of treatment is important. But if a patient has neurocognitive, neurobehavioral symptoms for 10, 15, 20 years, whether a 6-month treatment with an mTOR inhibitor is sufficient or not. And whether our negative findings might be due to the age of the patients that we treated. So I'll come back to that point in a few minutes. So what I wanted to do is to highlight, again, going back to our clinical studies, how unique TSC is. A lot of times, individuals with autism do not get diagnosed till they show behavioral symptoms until 12, 18, 24 or older months or older. But in TSC, what we have is a sort of prenatal or neonatal test due to the presence of these cardiac tumors. As I mentioned to you earlier, among fetuses and newborns with multiple cardiac tumors, the chance of having TSC is 95%, which is actually higher yield than doing genetic testing. Given that high incidence of TSC, and given the fact that half of these patients are likely to develop autism spectrum disorder, we wanted to ask the question, do we know which patients will go on to develop autism and which ones will not? Is there a red flag? Can we detect which infants-born TSC will go on to develop autism? There are not that many genetic disorders where you can do this kind of analysis. So we've been focused on this question for many years. started with Simon Warfield, Shefali Jesti, and Chuck Nelson here at Boston Children's at a single site. We did neurocognitive assessment, diffusion tensor imaging, MRI, and neurophysiological assessment of visual paradigms. I'll show you some early results that we published almost 10 years ago, or more than 10 years ago now, where we looked at corpus callosum as a region of interest and did diffusion tensor imaging in non-TSC patients, TSC patients that are not on the spectrum, and then finally TSC patients that have autism spectrum disorder. And as you can see in these visual representations, the control patients without TSC and TSC patients that are not on the spectrum seem to have very similar corpus callosum organization. And in fact, we can use some quantification such as fractional anisotropy, which is a measure of the microstructure of the corpus callosum. And the fractional anisotropy in these two groups is very similar to each other. However, TSC patients with autism spectrum disorder seem to have much less organized corpus callosum. And this might be related to axonal connectivity or myelination, but seems to be an important correlation here. So this was a single site study of about 40 patients. So we wanted to validate the results using larger study. So we formed a Autism Center of Excellence research network called TACERN, and identified patients born with TSC from these 5 sites around the country. And I'll share with you some of the results. I should say we've confirmed the imaging findings that I just showed you in this group of patients as well. But we also started looking at EEG as a potential biomarker in TSC. And what we did was to do prospective longitudinal EEG acquisition in patients born with TSC. And what we found was that EEG is often normal in the babies born with TSC, shown by these white bars. The EEG becomes abnormal with interictal spikes, shown by these gray bars. And then finally, you get the presence or the onset of clinical seizures indicated by the diamond figures here. So we collected data from a small subset, and the finding was that the first time of epilepsy from discharges an abnormal EEG is around 4 months of age, but these patients don't have clinical seizures at the time. The clinical seizures start around 6 to 7 months of age. So we have a 2 to 3 month period where the EEG is abnormal, but the seizures have not started clinically. This led to a trial called PREVENT, led by Martina Bevan at Alabama. We are using vigabatrin in infants born with TSC. And there's an early vigabatrin treatment group versus a delayed vigabatrin treatment group. Delayed vigabatrin treatment basically starts when the clinical seizures start. So we're asking the effect of pre-clinical seizure treatment with vigabatrin, which is an anti-seizure medication, on development. When we proposed this to the FDA, they wanted us to focus on development at 24 and 36 months rather than the seizures as the primary endpoint of this treatment. So this study just finished, and the results will be publicized soon. I should say a similar study was done in Europe already called EpiStop, and they showed some improvement in seizures, but not in development in that study. So we're anxious to share our results soon, hopefully within this year. There are some differences in the design of these studies. So it'll be interesting to see what these two studies say about the use of pre-symptomatic, if you will, or asymptomatic treatment with an anti-seizure medication in TSC babies. This idea of prevention rather than symptomatic treatment, I think, is a very important concept in child neurology, and it will be important in other diseases as well. Now here we're using vigabatrin, which is a GABA transaminase inhibitor, which increases GABA in the brain. But obviously, many of you are thinking, why won't we use an mTOR inhibitor? And we are actually doing that using an FDA-funded study led by Darcy Kruger. It's very similar to the PREVENT study that I showed you earlier. It's ongoing at 8 sites currently for babies born with TSC. And we're using sirolimus or rapamycin to see if we can delay the seizures or prevent the seizures in babies born with TSC. So I'll conclude here. I've talked about one or I guess 2 genes, TSC1 and TSC2, and one disorder, tuberous sclerosis, in this talk. There are roughly 900 plus other genes that are similarly implicated in neurodevelopmental disorders. How are we going to go after the bigger landscape of these disorders? So we've actually formed consortium that is looking at 3 genes comparatively: tuberous sclerosis group of patients, P10 patients with P10 pathogenic mutations, or P10 hamartoma tumor syndrome patients, and then patients with SHANK3 mutations, or called Phelan-McDermid syndrome. This is a study that's been going on for 10 years now. We're collecting patients from 11 sites, and we have biomarker identification such as MRI and EEG, as well as phenotyping of these patients. The idea here is that we're focused on the mTOR pathway as a potential convergence point, with P10 and TSC potentially upregulating mTOR and SHANK3 loss downregulating mTOR, and looking at whether we can start to identify differences between these different disorders and look for convergent potential intervention arms. So we've actually done a mTOR inhibitor trial in P10, and that looks quite promising. We just published that last year. We have more trials coming up the pike for these disorders. I will stop here, acknowledge the work that I presented by our various lab members, and I should acknowledge that this would not have been possible without the collaboration of many investigators in the field, and I'm grateful for our funding sources. I will stop here and answer your questions. Thank you very much for having me.